Okay. So, so what I'm going to do is I'm going to give a quick introduction to dkNET and some of the tools that we provide. As Marianne had mentioned, we're funded by National Institute of Diabetes and Digestive and Kidney Diseases, and really, dkNET is meant to be a portal for research resource information for biomedical researchers. And when we're talking about research resource information, that will become a little clearer as I go through some of the slides. But really, different types of information, from information on resources you may use, like antibody cell lines, data sets that you may want to have access to, and then also information on sort of rigor and reproducibility guidelines from NIH. How does one comply with that, or how does one comply with data sharing mandates coming out of NIH? And really, it's building out sort of this information network as well, so that we provide this information to researchers, but we also try to help connect researchers in the field with other NIH-funded centers as well. So there are a number of centers that are funded by NIDDK that also provide resources that are useful in your research. And as we mentioned, we are a laboratory at UCSD, and the Fair Data Informatics Lab that Marianne and I co-direct supports a number of major informatics projects in biomedicine. So dkNET is one of them. We're also involved in development of some data repositories for, as I mentioned earlier, the spinal cord injury and traumatic brain injury domain. We're also involved in other areas, developing data curation, knowledge management platforms, and the like. So, we're involved in a number of these large programs. So, in terms of the 5 major areas in the portal, and we'll walk through each one of these. First one is the resource reports, and this is really to help researchers find and evaluate resources for their research. The authentication reports are really there to assist in preparing NIH authentication plans, and also tracking the resources that you are potentially using and seeing if there are any updates in terms of any problematic reports about them, or any other information about these resources. We also have a set of services around fair data. This also, this includes finding data repositories, but also providing information on preparing data management plans and the like. Discovery portal connects you to a number of biomedical databases that you can search through, and then the Hypothesis Center is really trying to provide powerful data mining tools to the researcher based on a number of omics data that are publicly available, but doing it in such a way that even bench researchers, so you don't need any sort of specific bioinformatics expertise, but basically providing a platform that allows you to easily go in and explore for example, genes of interest, pathways they might be involved in, and the like. Okay, so when one looks at sort of the entire sort of a research workflow, right? So the tools that that dkNET has really support a number of different areas. Everything from the background research, where you can use the discovery portal to find perhaps preliminary data, funded grants already working on a similar area, or potential funding opportunities, to developing a hypothesis. So for example, you could go into the Hypothesis Center, and again, Hypothesis Center is going to be a full lecture this week with Dr. Neil McKenna walking you through how to use that. And then you'll also have the opportunity to work with SVP, and we'll have a follow-up workshop day where we'll further work with you in the Hypothesis Center. Once you sort of, have your hypothesis, when you're planning your experiments, finding the appropriate resources, finding, the appropriate antibody or cell line, seeing, how others have used it, linking to, publications or reports about that. And then also, developing your authentication report. So this is something required by NIH, all the way to collecting and analyzing your data, where again, you may want to go into finding, for example, through the resources, finding specific analysis tools for certain types of data that are used in the community. And then when you're publishing your results, the information we have on fair data resources, and also, when you're publishing, ensuring that you're properly citing, the resources that you've used. When you come to the dkNET portal, I mean, those main areas, as you see, are sort of outlined on the top. Those take you into those specific areas. So getting more in depth into the resource reports. And again, this is, where you're, can be used very beginning when you're planning experiments, all the way to through the publication, when you need to find the research resource identifiers associated with that. So, the resource reports themselves are, each resource itself has its own specific page that provides information on the resource. We have information on, in this case, the antibody itself. We have information on usage and citation of that resource. The information we're able to provide in the usage and citation metrics is the citation and usage in the open access literature, and the open access literature that we're able to actually text mine. Then we also have other information on ratings and potentially alerts about the resource. And if you go through to the usage and citation metrics, you'll see information on, how often, that resource is mentioned. We have links to the papers, that mention that resource. And we also provide, a listing of other resources that are frequently cited with this, with this particular resource. So, really trying to provide, information for you to find, out more about, the specific antibody in this case, or, could also be a software tool, could be a cell line. And this is what we call, the resource information network. And again, this is going to be covered more in depth tomorrow, where we talk about rigor and reproducibility and RRIDS. But really, this information network is trying to assemble, information about, these key research resources from various areas. So, as we pointed out, from pointing to the literature, sources of information about that resource, information provided by other projects. For example, the Human Protein Atlas provides validation information for example, a number of antibodies. And the Cell Line Authentication Committee provides information, about cell lines and, if, for example, a cell line is contaminated or not. And all this information is tied together through these research resource identifiers. So once we have an appropriate identifier for a resource, such as an antibody or cell line, we can then start attaching, this other information to it. And so, what are research resource identifiers or RRIDs? They are the persistent identifier for research resources. And, we were, instrumental in development and adoption of the RRIDs. So this goes, back a decade in terms of, beginning to, work with, publishers to better identify resources in the literature. Because, 10 years ago when we started, working on this, it was very difficult to actually go into a research paper and then a lot of times track down the specific antibody or the specific cell line that was found or that was mentioned in the paper. And so, what we've worked on is the development of this identifier, and it's really meant to help in defining what resources were used in the study, and then, as I mentioned, linking this ID to other publications or other information about this. And these RIDs are supplied by the authors when the paper is being published, and it's part of the materials and methods section. And this is now incorporated in a number of journals that require them. Many journals recommend them, and they're also built into a number of reporting guidelines, such as STAR methods. And the growth of RIDs has been pretty significant and continues to grow. As I mentioned, it's used across, basically all, the major publishers. It's also part of the standard that, for example, the National Library of Medicine uses when they get the abstracts from the various journals to display in PubMed. Most recently, the material design analysis reporting checklist, something that Science and others are picking up, and it's included in the MDAR checklist. There are more than sort of 74,000 distinct resources that are identified in journals. And one of the key things that I mentioned that we're tying into are the ratings and alerts. So as I mentioned, there are a number of, for example, ratings provided by different groups. So for example, this is the Intestinal Stem Cell Consortium, for cell lines. The International Cell Line Authentication Committee, provides the feedback on problematic cell lines. And I, mentioned the Human Protein Atlas. And so, by being able to aggregate this information around RIDs, from multiple sources, we're able to aggregate that and provide that information, when you're looking at the resources, that you're working with. So, once you've chosen, a number of resources that you'll be working with, we also provide the ability to collect those in a group and then help you in, developing authentication reports. And so as part of NIH's Rigor Reproducibility guidelines, there are a number of mandates in terms of what you must supply, with your research proposals. Part of that is, the authentication of key biological and chemical resources. And so, that includes cell lines and antibodies. And so providing, information on what methods, you're using to authenticate these resources, but also, providing a means for you to, track, the information about these resources over the time of your award. Another thing that NIH does require is the data management plan, and NIH just released its new data sharing, sort of its final data sharing guidelines, which will be implemented in 2023. And so we'll discuss this on Thursday and Friday when we talk more about fair data and data management. But NIH is now in 2023 going to expect all funded research proposals to make their data available, to the broader research community. And so that, will definitely be something, that's important, as you're setting up your research plan, working through, your research process, figuring out what you're going to be doing with your data and, how to actually then, make that data available as per NIH guidelines is going to be a critical part of your planning, and something that we'll cover in terms of describing some of this. And so with the authentication reports, what we provide, as I said, there are two things that we provide. So you're able to, create a collection. For example, here's a collection of cell lines, and you'll notice that one of them has a little red triangle next to that. And this is, in this case, it's because it's a discontinued antibody. So what we're trying to do is, allow you to collect, sort of group your resources that you're using. We provide information on different validation techniques, and this is information, from the community, that is, developing, these various validation criteria. So basically providing, the information and the resources, for you to look at that. But then, as you're doing your experiment, these authentication reports are live in the sense that, you are able to come back 6 months later and see if there are any, alerts related to, the resource or resources that you are currently using, and you're actually able to snapshot sort of the authentication report, by generating a PDF, of the report at a specific time. So this provides a way for you to sort of again track, the usage of your resources. So the next thing I want to cover is the discovery portal. And here again, we provide search across a number of different resources. And so, if you go in and search, for example, diabetes or something else you might be interested in, we break that down into a number of different information resources that may exist around that. We have subcategories that you can filter on, and then, takes you into, some of these resources. Now, again, we're not funded like Google, so we only crawl these resources weekly, bi-weekly, or monthly. However, for a number of these resources, we can sort of index them at a slightly deeper level in terms of extracting some of that information, and provides a nice place for you to then actually also filter the type of information you might be looking for. So instead of getting just a list of papers, you can look for things related to genes or models or phenotypes or organisms, for example, related to your search. And one of the things that we do as dkNET is that we do bring in a number of DK specific resources that you can search across. So, for example, the mouse metabolic phenotyping centers, IIDP, DICOMP, and then again, some of the more common organism, antibody, cell line information is also available. And now when you're looking at, as I mentioned, sort of publishing data, we do have these FAIR data resources, and so if you click on sort of the FAIR data part of that, there's a number of different pieces of information. So we have some information on research data management, and then sharing data, where we provide suggested data repositories and link you to the standards that they use. And so again, this is be covered a bit more later in the week. And for research data management, there are a number of tools that are available. For example, for a data management planning tool, for example, provided by the California Digital Library. So we point you to some of that information through these pages. And then where can you deposit your data? We do have the listing of data repositories, and when you go through the list, there is sort of a breakdown by where the support for this. So is it an NIDK resource or an NIH resource or another institutional repository? We also have scientific disciplines. So, for example, if it's proteomics data, what are the data resources, data repositories that accept proteomics data. And then along with each data resource, you'll notice that there are a number of different icons that can give you information on, for example, if you want to upload data to the resource or download data, information through fair sharing. So fair sharing provides information on what standards a resource uses. And so, we try to connect you to, relevant information about a resource repository that you might be interested in. And then, the Hypothesis Center, we've mentioned, really looking at, genes and, how they're related to signaling pathways. And so this will be, the lecture during the boot camp on June 23rd. And, really the Hypothesis Center, and SVP, is meant to, connect bench researchers to data, that is already out there, but through SVP has been curated to make it more fair and to allow sort of data mining across all the different omics data, that's provided by resources such as dbGaP and others. And so, you can find genes with important roles, related to receptors, enzymes, organism tissues, and also explore and define signaling pathways, relevant to a single gene or regulation. So, it's a really nice tool to be able to go in and explore, these various attributes, and Neil will guide you through that. And he'll also, as I said, we'll have the follow-up workshop. So if you're using, if you want to use SVP to start exploring, some of the, genes or receptors or signaling pathways that you're involved in, Neil will also be available, to assist you over the summer program. For example, from last year, there are already, a number of students, who used SVP who have now, gone on to publish some papers, where SVP was involved. And so, stepping into a summary again, so really, if you're, walking through a specific question, such as, perhaps, you're interested in patients with diabetes and how this relates to problems, with the eye. You can, again, SVP, can be used to look at, potential signaling pathways, genes related to insulin receptors, such as PMAI-P1. Questions come out of that, do the genes that regulate, the insulin receptors in islets also, regulate receptors in the cornea. You then have to, get to, doing background research, if, insulin receptors in the cornea. So there are a number of, papers in the literature around this. But, if you're looking at, PMAI-P1, you may want to find then appropriate antibodies, to further, look at that protein. So using, the resource reports to find appropriate antibodies or cell lines. You've collected, your imaging data. You may then want to find, tools, that actually, help you quantify image intensity. And so, there are a number of tools, that are represented, through the resource reports and the software registry, that you could potentially use. And then, once you're, done with your study and you're going to publish your results, then you'll need to bring back, the research resource identifiers for the analysis software for the antibodies and cell lines, but you'll also need to find, because of the 2023 mandates, you'll need to find, the appropriate repositories for you to deposit that data. And so that's sort of the overview of the tools within dkNET. We through the homepage, there are a number of other areas that where we provide information and also resources. So we do have a webinar series, and so the webinar series covers a number of tools and resources available to you that might help you with your research. These are all stored on our YouTube channel, so you can go and view prior webinars, and the slides are also made available through SlideShare in case you just want to look through the slides themselves. But this is something that we will continue to do. The next series will kick off in fall this year. And we do, we've been focusing a lot on some of the informatics tools, that can help, with research and with exploring, areas within a field. We do also have a newsletter, so we provide, community news, new funding opportunities, events, where people, can sort of reach out to us. And we have a sort of a virtual booth where you can, check out all the resources. And then, we do have, as I mentioned, these also these news releases that we put out about other centers that may have important information, funding, and other, for example, educational opportunities, and then the mailing list, which will provide information sort of on a monthly basis.